Welcome to my presentation on electron microscopes and light microscopes. Some key definitions that we need to know is that resolution is the ability to distinguish between two very close points. The greater the resolution, the more clear an image is. Magnification is the number of times an object can be enlarged. Light microscopes tend to use wavelengths of light to be able to produce a magnified image. This is done by a light source under the stage of the microscope shining light on the glass slide containing the specimen. The light will travel through the specimen and then through a number of lenses which help to enlarge the specimen. The problem with light microscopes is that they have a relatively low magnification and resolution. On the other hand, electron microscopes are also used to magnify a specimen, but they use electrons. Electron microscopes have a higher resolution and magnification than light microscopes, as they use shorter wavelength electron beams to magnify the specimen. The negatively charged electrons are focused onto the specimen using an electromagnetic condenser. Due to the fact that the microscopes contain an electron beam to magnify a specimen, the electron microscope must be in a vacuum in order to prevent the electron beams colliding with the air molecules. There are two main types of electron microscopes, the transmission electron microscope and the scanning electron microscope. So the TEM basically consists of an electron gun, which fires an electron beam that would be focused onto the specimen by an electromagnetic condenser. The electron beam will be fired from below the specimen, so below the slide or the stage. Some parts of the specimen will appear more dense than others and therefore will absorb more electrons, thus appearing lighter, and a 2D image will form. The TEM often has a very high resolution, but this can be compromised by the difficulties preparing the slide due to the very complex staining process required, and this may cause the formation of artifacts, therefore hindering resolution. Also, high electron energy beams may also destroy the specimen, therefore meaning that the resolution capability might be quite limited. And it will also affect the magnification that you can go to, because the higher the electron beam, the less you'll probably be able to see because you may destroy more of the specimen. Another problem with a transmission electron microscope is that you can't use living specimens to observe, as the whole system must be kept in a vacuum, and therefore you'd kill off any living specimen that you placed in because they wouldn't be able to breathe or respire. The specimen must also be very thin in order to allow electrons to pass through, which again is another limitation. On the other hand, the scanning electron microscope is similar to the TEM in the way it operates. However, the scanning electron microscope will direct a beam of electrons onto the surface of the specimen from above rather than below. The beam then is scattered across the specimen according to the contours of the specimen, and the pattern of the scattering will be detected and used to help to develop a 3D image of the specimen. The SEM microscopes have a far lower resolution and magnification than the TEM microscope, but they still have a far greater magnification and resolution than the light microscope. Some key formulas that we need to remember when doing microscope calculations is that magnification equals size of the image divided by the size of the real object. And also the other equation that we need to know is the total magnification equals the eyepiece lens multiplied by the objective lens. If you found this presentation useful, don't forget to subscribe and give it a thumbs up. Thank you for watching.